Yes, students, we are live. Awesome. It took us some time, but we are live right now. Yes, am I audible? Yes, students, we are live. Awesome. Okay, voice is also clear. Yes, I can see your chats as well. So all of you can send me a hi. Now let's start every single line of NCERT. Okay. Chalo. Now what is this locomotion and movement? Now students, what makes a living and non-living thing, separate? What do, like from smaller class we are learning that living organisms have the ability to move, non-living organisms do not have the ability to move, right? These are the simple things that we have learned. So in this chapter, what you're going to basically learn, you're going to learn how are we doing the movement, okay? How our muscles are contracting, how they're relaxing, who are the proteins that are helping behind this, which are the different types of bones, and what is our axial skeleton, appendicular skeleton. All this is what you're going to learn in the class today, okay? And we're also going to learn how are there some normal locomotions that are present, okay? Just give me 1 minute. Yeah, which are the normal movements that are seen in our body, and how do they contribute for our day-to-day activities. Like now when you're trying to contract your muscles, you're not thinking much, right? You're not thinking, oh, I want to contract my muscle after an hour, will my body do that? You're not thinking all of that. You're just casually doing it. Right now I'm doing this, I'm not even thinking. So that means what? They are in my control, and it happens in a very less time, right? It's happening in a very less time. So let's see what's the first few statements of NCERT. Movement is one significant feature of living beings. Animals and plants exhibit a wide range of movements. Streaming of protoplasm in a unicellular organism like amoeba is a simple form of movement. So students, all of you know amoeba is your unicellular organism with hardly any shape, right? There'll be something like this. Okay, let's say, so protoplasm is cytoplasm plus the nucleus. So when they are moving with the help of your pseudopodia, what is going to happen? That streaming of protoplasm is going to happen, or movement of protoplasm is going to happen. If this is happening, that is also one kind of movement, yes or no? Now same way, when we learn animal kingdom, we learned about tentacles in your coelenterata, and these tentacles had your nidoblast. This nidoblast cells would be released to capture the prey. All these are the small movements that is done by the organism to survive and to live happily, okay? So yes, amoeba, simple form of movement of cilia, flagella, tentacles are shown by many organisms. Human beings can move their limbs. Yes, we can move their limbs, we can move our jaws, we can move our eyelids, we can also move our tongue, okay? Some movement result in change of place of location. Such voluntary movements are called as locomotion. So students, let's say there is Chintu, okay? This is Chintu, okay? This is Chintu, and this Chintu is going to the shop, okay? This is the shop. Now if Chintu is walking, right? Chintu is walking and he is doing displacement and he has reached the shop. That means I will call this a locomotion, okay? I will call this a locomotion. But imagine at the same place we have Mintu, okay? We have Mintu. Mintu is standing and Mintu is raising her hands, okay? Raising her hands. Then Mintu is standing and she's keeping her hand straight, okay? She's keeping it in 180 degree. Then she's standing in the same place, okay? I'm just doing different emojis. She's standing in the same place, lifting only one arm, and one arm is down, okay? Now all this is called as movement, okay? This is called as movement. Why this is called as movement? Because of a simple factor that displacement is not happening, okay? No displacement, okay? No displacement. When there is no displacement, you're standing in one place and you're doing the activities. Like when we do it in aerobic, students, we used to all go for our PT class, no? What our PT teacher used to make us do? Make us stand in rows, and in that rows they used to ask us to lift our hands, okay? Do something like this, that is movement. But locomotion is what? When you displace, that means you go from one place to another place, okay? When you displace, that is called as locomotion. Is this clear to all of you? So here there is few statements that you have to remember, so that's why I'm stressing on these two words. Now walking, running, climbing, flying, swimming are all forms of locomotory movements. Locomotory structures need not be different from those affecting other types of movement. For example, in paramecium, cilia helps in the movement of food through the cytopharynx and in locomotion as well. Hydra can use its tentacles for capturing its prey and use them for locomotion. So students, Paramecium has cilia. All of you remember it is, it will look like the sole of your shoes, right? Like this, something like this, and something like this. Okay, this is how your paramecium is going to look, and they'll have full cilia. Okay, everywhere cilia will be. Cilia, 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 cilia. Okay, everywhere cilia is going to be there, and this cilia is going to help in the movement of food through the cytopharynx, as well as it is going to help in locomotion. Now hydra also, students, hydra have tentacles. Okay, this is your hydra. Okay, many tentacles will be there. I'm just drawing a very mediocre hydra. Okay, this tentacles will be there, many tentacles will be there around the place. Okay, this is going to be smaller structure. This tentacles is going to help them in capturing the prey, as well as going to help them in locomotion. So you can understand that there are different types of movement happening in every organism, and these movements actually help the organism not just for locomotion, but also like capturing of its prey, or feeding themselves, all this, or passing the food from one place to another. Now we use limbs for changing in the body posture and locomotion as well. The above observation suggests that movements and locomotion cannot be studied separately. Okay, that means let's say I'm trying to do locomotion. In that case, when I'm doing locomotion, there are many movements that I keep also, right? So the two may be linked by stating that, okay, this is a very important statement. All locomotions are movements, but all movements are not locomotion. Yes or no? Because in movement, it's not necessary displacement happen, but in locomotion, displacement is necessary. So methods of locomotion performed by animals vary with their habitat and demand of situation. So two things are going to affect the locomotion and movement. That is going to be the methods of locomotion. Let's be correct. Okay, methods of locomotion. So methods of locomotion, students, two things are going to affect. Okay, first thing is going to be habitat. Okay, first thing is going to be habitat, and second one is demand of the situation. Demand of situation. Let's say Mintu is walking and there is a dog running. Okay, dog is coming to bite her. Now will she walk? The demand of the situation is to run, so she will start running. Okay, she'll start running. Imagine she's walking with her grandmom. Okay, she's walking with her grandmom, and obviously grandmom is old, so grandmom is walking very slow. Okay, grandmom has a stick and grandmom is walking very slow. Now what Mintu will do? Mintu also will start walking slow. That is the demand of the situation. So same way here, demand of situation means how these organisms are, where are they located, how is their surrounding, are they in the marine form, freshwater form, terrestrial, land, flying, depending on all of this, and depending on what situation they are going to be in, all that is going to matter. Okay, all that is going to matter. Clear or not? Okay, methods of locomotion performed by animals vary with their habitat. Okay, vary with their habitat and the demand of the situation. However, locomotion is generally in search of food, shelter, mate, suitable breeding grounds, favorable climatic condition, or to escape from enemies. Now you tell me, why do we go to work? We go to work so we can buy food. Okay, we go to work so we can have our own shelter. We go to work so we can have a good partner for us. We go to work. Now for animals move around for getting suitable breeding grounds. And why do people migrate? Why do people go from one place to another place? Now people who are staying in Bangalore is migrating to inner cities, or people who are in the inner cities are migrating to Bangalore. Why? For the simple fact that climatic condition, the job facilities, employment, and studies for their kids, all this education for their kids, that's why they are migrating. So all this is why we do locomotion, or for this sole reason is why we do kind of locomotion, right? Locomotion and movement. I hope it was clear. Introduction, that is the introduction. Okay, now types of movement, students. Namely, there are three types of movement. Okay, mainly there are three types of movement. First one is called amoeboid movement. Okay, amoeboid movement. Now before I go into each of the movement, you should understand these are the movements that is happening in our body as well. See, amoeboid movement is happening in amoeba, no doubt in that. Amoeba is doing the movement. Like, ma'am, I know about amoeboid movement, right? This same amoeboid movement is happening in our body also with the help of WBCs and macrophages. Okay, with the help of WBCs and macrophages. These people, that is especially your WBC, how do they do amoeboid movement? Is because of diapedicis. Because of diapedicis. Diapedicis means what, students? Okay, look here. This is your capillary. Okay, this is your capillary. I hope voice no problem. Amoeboid movement. Okay. Okay, now this is the capillary. Now let's say I don't think I'll be able to do this, so let's do another trick. Okay, let's say that these are the small pores, okay, on the capillaries. There are small pores on the capillaries. Okay, small pores on the capillaries. Now your WBCs are going to, students, this is your WBC, they are going to ooze out of this capillary. Okay, they're going to ooze out of this capillary and they are going to go outside and they're going to do phagocytosis. Now ma'am, you can ask me a question, ma'am, why are they doing this? Students, it's very simple. Let's say here there is a tissue. Okay, here there is a tissue. Okay, this is our tissue. Okay, tissue. This is the tissue. Now we notice that in the tissue there is a disease causing agent. Okay, there is a disease causing agent. Let's say here there is a disease causing agent, and we are noticing, the WBC have noticed that this might spread. Okay, this might spread on a long run. So what WBC is going to go do, it is going to go all the way, it is going to squeeze out of the capillaries and it is going to engulf. Okay, it is going to engulf this fellow and take this fellow and do phagocytosis. Okay, so he is going to be engulfed inside the WBC and they are going to do phagocytosis. Understood? So for this phagocytosis to happen, this process is called as diapedicis, where through the pores of the capillary, the WBC is going to squeeze out and it is going to go and attack the cells which are having problems, or tissues where we noticed a problem. So amoeboid movement is shown by WBCs and macrophages. Clear? Next one is going to be ciliary movement. Ciliary movement. Ciliary movement, students. See, ciliary movement means cilia. Yes or no? Cilia. So that kind of movement happens in the lining of your fallopian tube. Okay, lining of your fallopian tube. Okay, lining of your bronchi. So in these places, what is going to happen? These are the tubes. Okay, these are the tubes that are there. Now inside this tube, there is going to be your there is going to be ciliated columnar epithelium or cuboidal epithelium. Now these people are going to do the movement. Okay, what kind of movement are they going to do? They are going to help in pushing this person, telling that you go that side, this is where you are supposed to go. If ovary is coming, fallopian tube will tell, please go this way. Okay, same way if air is entering. Okay, if air is entering, then trachea and bronchi will tell, please go this way, this is where you are supposed to go. So in the lining of the fallopian tube, lining of the trachea, bronchi, all these places, this person is present. Who is that? Your ciliary movement we can see. Okay, ciliary movement we can see. Next is going to be muscular movement that we are going to learn in this chapter. Muscular movement, students, how many muscles do we have? Around 639 muscles we have, and in these muscles only are striped muscle, or we call it as striated muscles, or we call it as the main word is skeletal muscle. Skeletal muscle is going to be the one who is going to be voluntary, who is going to listen to us. Okay, who is going to listen to us. Clear or not? Clear. Okay, I'm just the background is dark and clean. No? Okay. Okay, done. So some of the specialized cells, see here they have given, okay, some of the specialized cells in our body like macrophages and leukocytes in the blood exhibit amoeboid movement. As I told you, this one WBCs, and both of them are going to do, you know, amoeboid movement. Okay, it is affected by the pseudopodia formed by the streaming of protoplasm in your amoeba. Cytoskeleton elements like microfilaments are also going to be involved in amoeboid movement. Okay, microfilaments, that is your actin. Okay, actin also is going to be involved in your amoeboid movement because of the sliding. Now, ciliary movement occurs in most of our internal tubular organs, which are lined by ciliated epithelium. As I told you, they will have columnar type of ciliated epithelium, or they will have cuboidal type of ciliated epithelium. So when they have this kind of cilia, what is going to happen? It is going to tell, please go this way, please go this way, please go this way. All this dance they will do. Okay, now the coordinated movements of the cilia in the trachea helps us in removing dust particles and some of the foreign substances. Okay, dust particle, and for telling that substances, that removing dust particles and some of the foreign substances. So this so students, whenever, like, let's say hair inside your nose, okay, all of you touch your nose and see you will have hair inside the nose. Don't touch inside the nose, just know and understand. You all have seen in olden people also. Now that hair is going to allow, what are they going to do? They are going to filter the dust particles. So when you're inhaling, there's a lot of dust in the, you know, in the atmosphere. So when you're inhaling that, what is going to happen? They are going to filter the dust particles. Okay, going to filter the dust particles. So the coordinated movement of cilia in trachea helps us in removing the dust particles and some of the foreign substance inhaled along with the atmospheric air. Passage of ova through the female reproductive tract is also facilitated by the ciliary movement. This is what I told you, lining of fallopian tube. Okay, lining of fallopian tube. That. Okay, now muscular movements. Okay, movements of our limbs. Okay, our skeletal muscle is the one who is going to help us in the movement of our limbs. It's going to help us in the jaws, tongue, I mean not the bones, I'm telling other ways. Okay, they're going to help in the movement of the jaw, tongue. So require muscular movement. The contractile property of muscles are effective used for locomotion and other movements by human beings and majority of the multicellular organisms. Locomotion requires a perfect coordinated activity of muscular, skeletal and neural system. Now imagine students, you are planning to run. Okay, dog is coming to bite you, you're planning to run. Your first thing will be your muscles have to move. They have to contract and relax for you to run. So again, muscles have to contract and relax. At the same time, your skeletal muscles, the bones also have to help you for that. Then the third thing is your neural system. First, your neural system will send you a signal telling that, you know what, you are in an emergency situation, you are supposed to run. After the signal is taken, your muscles are going to contract, even your bones are going to help and favor them, and that is how your entire body is going to be locomoted. So locomotion is going to happen when your muscular, okay, skeletal and your neural systems are working. Okay, neural systems are working. In this chapter, you will learn about the types of muscles, their structure, mechanism of their contraction, and important aspects of their skeletal muscles. Okay, hi Panda. So here, always remember, okay, always remember that you need to take the help of your muscles. Okay, muscular help, muscle help you have to take. Then you have to take of your skeletal muscles who are going to listen to you. Okay, other two are not going to listen. Who is that? Smooth muscle and cardiac muscle will not listen. Okay, so don't even expect them to listen because they are involuntary to your body. Okay, only your skeletal muscles are going to listen. And then you have, who you have, muscular is there, skeletal muscle is there. Then you're going to have your neural system who is going to respond and tell that, hello, please run, otherwise the dog is going to come and bite you. Or when you're touching a hot object, okay, who will ask you to retrieve your hand? Your neural muscle, students. Neural muscle is going to tell, please retrieve your hand, or else your hand is going to get burned. Right? That is what we learn in reflex action. No? What I'm talking now is what I'm teaching you in reflex action. Reflex action. Done. All of you, yes ma'am, done. Okay, reflex action. So friends, so far I think I'll just write it here. So amoeboid movement, is going to happen in what? If I write in this corner, are you able to see? Yes, very good, Panda. Neuron. Okay, I think you can see, no? Wait, I have to wait for this to load. It is seen. Okay, so amoeboid movement is shown by which type of cell, students? Amoeboid movement is shown by two types of cells. That is going to be your WBC, and WBC is doing the movement because of diapedesis. Okay, diapedesis, that is squeezing out of the capillaries to protect the tissues, okay, which are affected. So WBCs and your macrophages are going to do amoeboid movement. With that, they have mentioned about the cytoskeleton. Okay, cytoskeletal, that is your microfilament. Okay, microfilament. Okay, microfilament. I think it's visible till there. Okay, microfilament. Next is going to be, after amoeboid movement, is going to be ciliary movement. Okay, ciliary movement. Which are the organisms that do ciliary movement? Your paramecium is going to do ciliary movement. That is why we talk about it under cilia. Okay, now under ciliary movement, you have to remember the example of lining of trachea. Okay, tracheal lining of the trachea, and bronchi. Okay, lining of the trachea and bronchi. Then lining of fallopian tube. Okay, lining of fallopian tube. And the third one, you can remember ventricles of the brain, but it's not mentioned. So this two lining is enough. And lining of fallopian tube is going to help the ova. Okay, it is going to help in the movement of ova. Okay, movement of ova. Clear? Then next one, muscular movement. Muscular movement, it's going to be your limbs. Okay, jaws and tongue. These people are going to do muscular movement. Okay, yes, I remember you, Mohit Raj. Okay, so much hangama you did in last class. No, how will I forget you? Clear, no? So till microfilament, I think it's visible. After that, till it's getting cut. So students, flagellary movement is there, but it is only a sperm. Okay, only one example is there. Flagellary movement. Okay, done. Chalo. You have studied in chapter 8 that cilia and flagella are outwards of the cell membrane. You would have studied in cell chapter. Flagellary movements help in swimming of the spermatozoa, maintenance of the water current in the canal system of the sponges, and in locomotion of the protozoans like euglena. So the only example that you have to remember under flagellary movement, okay, flagellary movement is going to be your sperms. Okay, flagellary movement is going to be sperm. They are the only ones who are going to behave or have the same movement. Yes or no? So they have an acrosome, okay, the head region, the neck region, and they are going to have a long tail. So these are going to move exactly like your flagella. Okay, now here they're talking about your water canal system. So water canal system, like we saw in sponges, okay, that is what here we are talking about. Okay, just give me 1 minute, students. Yes. Sorry, one very important task came, that's fine. Sure. Okay. Next section. Now only I'm taking one session. No, next session you're already asking. Okay. Yes. Students, in Euglena also we have the flagella, right? So there is a short and a long flagella. So they are also going to help in the movement. Okay. Thank you so much. Wish you the same. About 40 to 50% of the body weight of human adult is contributed by muscles. So students, what happens? Why does a person become obese? It is because muscle weight will be reduced and the fat will be more. Okay, fat will be more. So that is why people go to gym and they build their muscles. Okay, all of them, all the boys will be like, yeah, I have triceps, I have biceps. Look at me, all of that. Why do they do? It's because of the fact that they have muscles. Okay, muscle gain has happened. Now they have special properties like, okay, they have special properties like, one second, I have to make this incident. Okay, done. Yes. Okay. Yes, students. Yeah, sure. So they have special properties like excitability, contractibility, extensibility, and elasticity. Which 12th chapter you want me to take? Just let me know. Okay, muscles have been classified using different criteria, students, like location, appearance, nature of regulation of their activities based on their location. Okay, based on their location, we have three types of muscle: skeletal muscle, visceral muscle, and cardiac muscle. Is this clear? Yes or no, all of you? Let me know in the chats. Which chapter you want me to take, just let me know. And students, I also have to tell you something. So on October 11th, without fail, join us on our channel, which will help you crack your boards, okay, as well as your KCET, Comet K exams. There is going to be a new channel for training you for your boards, that is your CBSE as well as your state board examinations, as well as for your KCET. Okay, the channel name is going to be Vedantu Boards and KCET channel. So definitely 11th is going to be a big launch, so please join, okay, without fail. Yes. Now students, here there have been 4 properties given about muscles. Okay, I will come to that. Before that, okay, here I'll just write it here. Okay, 4 properties of muscles. 4 properties of muscle. What are the 4 properties of muscles? Yes, tell me. Yes, just tell me the chapter name. Okay, I'll teach you. What are the special properties of muscle, students? First property is going to be very simple. Dog is coming to bite you. Okay, this example only I'm going to take. Dog is coming to bite you. Your neurons have sent you a signal. Okay, your neurons have sent you a signal. This neuron has sent a signal and muscles have to get excited. Okay, muscles have to get excited. Okay, one second. Mohit Raj, why don't you check out Vedantu NEET English? I have just taught recently genetics and biotechnology. You can check out the channel and you can definitely prepare. You do not have to wait for me to, reach till here. Okay, until I start here, you can definitely check out Vedantu NEET English channel. We have taken good lectures there. Okay, all the chapters I have completed there. Okay, just search this and you will find. Or you can just type Gopika ma'am biotechnology and you will find the classes as well. Okay, any one of this you can search. Chalo. Okay, now 4 properties of muscles. Very good, Panda. Excitability, ma'am. Excitability. That means ability of the muscle to take the action potential or to do the action potential. What do I mean by action potential? See, this is the muscle. Okay, this is, let's say, Panda's muscle. This panda's muscle is in a relaxed state, but suddenly the panda received a signal from the neurons, okay, from the axon terminal. Let's say here, okay, this is the axon terminal. They have received a signal telling that you are touching a hot object. Why don't you take your hands? So the first ever job of the muscle is excitability, the ability of the muscle to get excited and do the action for that. Okay. Second thing is going to be what? What is the second thing? Very good, ma'am. Elasticity. Okay, elasticity of the muscle, that means the ability of the muscle to stretch a little or to widen a little and then come back to its original position. Okay, elasticity of the muscle to provide that flexibility. Then we have extensibility. Okay, extensibility. Then we have contractibility. Contractibility. Yes. Oh, Casey. Okay. If you're in North, then it is not for you, dear. Okay. If you're in North, then you don't have to bother about it. Don't worry. Okay. Don't worry at all. It is not for you. It is for the Karnataka board. Okay. But you can take help for your board exam. Okay. You can take the help for your board exam. Clear? Yes. Okay. Okay. So elasticity means the ability of the muscle to, after it is getting stretched, for it to come back to the original position. Yes or no? Let's say if this is the muscle, okay, this is the muscle, if it has stretched, okay, stretching has happened, it has to come back to its original position. Okay, this is the original position, it will come back to the original position. Now extensibility means what? Extensibility means the ability of the muscle to get stretched. Okay, the ability of the muscle to get stretched and have that little flexibility. Then contractibility means what? The muscle's ability to come close together. Okay, close together, that is called as contractibility. Okay, so many hands are playing, no? No, you don't have to write KCET, dear, because you're from North. This is for Karnataka. Okay, this is for Karnataka board. Yes. Now, next thing is, students, the next topic that they have discussed here is based on location. Okay, based on location, there are three types. Okay, three types of muscles. Okay, Mohit, no use, dear. You're in North, no. So if you're coming to here to learn, that time you can write the exam. Okay, but otherwise you don't have to write. It is basically to get into good engineering colleges. Okay, yes, I'm teaching from Bangalore. Okay, I'm teaching from Bangalore. Okay, it's mainly written for engineering, but then students write it for agriculture, pharmacy. So that is your KCET exam. Okay, there's a common entrance exam here in Karnataka. So it's a Karnataka common entrance exam. So basically what happens is you write this exam for getting into good engineering, pharmacy, agriculture courses. Okay, agriculture courses. Now a person who has passed 12 and who has secured like 50% can write the exam, and then you have to pass in the exam. Okay, so you'll have chemistry, mathematics, biology and biotechnology. All these courses people can write the exam. Clear? Yes, all of you? Chalo, okay. Very nice, very nice. Okay, it's not for the medical students, okay? It's not for the medical students at all. So don't worry about the medical students, okay? So mathematics, physics and chemistry you will be learning in KCET exams, okay? Even biotechnology people also write it. Biotechnology, bio people also write it, okay? Because if they want to go for another field, they can choose between mathematics and biology. Okay. Chalo. Now, based on the location, yes, I'll be answering slowly about each of the exams. Okay. Now, based on the location, students, you have smooth muscles. Okay, smooth muscles. Then you have skeletal muscles. Okay, you have skeletal muscles. And you have cardiac muscle. Okay, you have cardiac muscle. Now just looking at the maybe the name, some you might be able to guess. Now smooth muscles, students, are the muscles that are present in the visceral organs. That means your stomach, kidney, all these organs are covered by your smooth muscles. Okay, smooth muscles. Skeletal muscles are the muscles that are present which are voluntary in activity. Okay, voluntary, and they will be connected to your bones. Okay, voluntary. Cardiac muscle, obviously it is going to be present in your heart. Now this muscle is going to be involuntary. Okay, involuntary. That means it is not going to listen to you whenever you want to contract. That is not going to happen. Here also, this is also going to be involuntary. Is this clear? Now we will go in depth for each of them. Okay. Oh, we came too down now. Yes, look here. We are going to start from each of it. First, you have skeletal muscles. You have to learn the characteristics, students. Same thing I have taught you in structural organization of animals. There also I have taught you this. So look here. Skeletal muscles are closely associated with skeletal components of the body. They have striped appearance. This is the first point to remember. What do they have? They have striped appearance. Striped. That means they'll have dark bands and light bands. So all of you would have seen muscle, no? This is how your muscle is going to look like, okay? And you would have seen that they have dark, light, okay, dark, light, like the striations will be there on the muscles, okay? This striations on the muscles is called as striped appearance. So they'll have striped appearance under the microscope, and that is why these people are called as striated muscles, okay? Striated muscles. Clear? Now, as per activities are under the voluntary control of the nervous system, voluntary control of the nervous system, they are known as voluntary muscles, okay? They are voluntary muscles. Clear? Voluntary muscles. Voluntary muscles. 1 second, students. Yes. Just give me 1 minute, students. Just lock this. Yes, clear? Okay, done. Okay. Now, so the first point that you have to remember about your skeletal muscle is going to be they are striped in appearance, okay? Striped in appearance gives them the name striated muscles, and I've seen it in many places that they will ask question about striated muscles and students are like, ma'am, which muscles are they talking about? Okay, so the answer is going to be skeletal muscles. Now, they are primarily involved in locomotory actions, and as per the activities are under the voluntary control, so they are under voluntary control as well. In locomotory actions and changes of the body posture, okay, changes of the body posture. So this person is going to help in the movement, okay, movement of our body. Next one is going to be visceral muscle. So students, visceral muscle or smooth muscles are two different names for this muscle. So don't get it confused. Don't be like, ma'am, they give smooth muscle, I did not know the answer for it. See, smooth muscle and visceral muscle are the same, okay? Smooth muscle is lining the visceral organ. Visceral muscle is also lining the visceral organ. That's why we call it visceral muscle. Visceral muscles are located in the inner wall. See, in the inner walls of the hollow visceral organs like your alimentary canal, reproductive tract, etc. They do not exhibit any striations and are smooth in appearance, so can hence be called as smooth muscles or non-striated muscles. Their activities are not under the voluntary control of nervous system and are therefore known as involuntary muscles. They assist, for example, in transportation of food through digestive tracts, okay, through digestive tract, gametes and through the genital tract. So here, what did they say? First thing they told, they are non-striated, okay, non-striated. That's the first thing they told. Second thing, they are involuntary, okay, they are involuntary. voluntary. Third thing that you know is they are not in the control of nervous system. Obviously, that's why they are called involuntary. They are present in the lining of the visceral organs. Okay, lining of the visceral organs. Lining of the visceral organs, and they are also going to help in transportation. Okay, transportation of food. Okay, transportation of food and gametes. Okay, and this visceral muscle or smooth muscle, they can also be called as that. Okay, next muscle you're going to learn is your cardiac muscle. Cardiac muscles, first thing is obviously they are muscles of the heart. Okay, muscles of heart. Now, many cardiac muscles assemble in branching pattern to form a cardiac muscle. So, students, they are branched. Okay, they are branched. Now, based on the appearance, cardiac muscles are striated. So, these people also are striated. This is why students get confused. Which one are they talking about? Mainly, they're talking about the skeletal muscle. Okay. Now, they are involuntary in nature as the nervous system does not control their activities. Obviously, can you control your heart? Okay, if it starts beating, if it starts, when you get panic and all, you cannot control anything, any regulation in your heart, telling that, okay, what heart block has happened, so I'll start pumping it. You cannot think like that, no? So, but in our skeletal muscle, we'll be able to decide, okay, I want to walk, I have to get up and go. So, that thought will be there, okay? So, these are your three types of muscles based on the location. Now, let us examine a skeletal muscle. So, before I go into the paragraph, let me draw and teach you. Okay. Oh, so much space I have left. Okay, student, this is the bone. Okay, this is your bone. Like Tom and Jerry, the dog had a bone, no? Similar. Like this is your bone. Okay, and it became too big. 1 second. Okay, this is the bone. Okay, now to this your skeletal muscle is going to be attached. Okay, to this your skeletal muscle is going to be attached. So this is going to be your muscle. Okay, muscle and muscle. Okay, this is going to be a skeletal muscle. Got it? This is going to be a skeletal muscle. Now, inside the skeletal muscle, students, there are going to be muscle bundles. What is going to be there? Muscle bundles are going to be there. Muscle bundles are going to be there. And inside this muscle bundles, okay, inside this muscle bundles, there is going to be muscle fibers. Okay, muscle fibers like this. Okay, muscles will be there like this. And inside this muscle fibers. So these muscle fibers are going to be long. That's why we're calling it muscle fibers. Okay, this is muscle fibers, they're long, so this is why we're going to calling them muscle fibers, because why? They're long and slender. This is called as muscle fiber. Okay, they are long, okay, and slender. That is why we call them muscle fibers. Now, these bundles, I told you, these bundles are called as fasciculi. Okay, bundles are called fasciculi. Got it? Bundles are called fasciculi. Now, let's say, students, these bundles are separated. No, bundles are all separate. To keep these bundles together, okay, to keep these bundles together, there is a material called, okay, there is a connective called as fascia. What is the connective called? Fascia or fascia. Okay, however you want to pronounce it. Then there are coverings of the muscle. Let's say this covering of the entire muscle. Okay, this is the first covering. The covering of the entire muscle is called as epimysium. Okay, epimysium. Epimysium. Okay, the covering of the entire thing is going to be called as epimysium. Okay, and inside, so that's the first layer. Okay, one transparent layer will be there. Then inside that, inside this bundles are there, no? For the bundles also there is going to be a covering. Okay, if I draw it in white, it won't be visible. Yeah, I'm drawing it in blue. This, okay, this muscle bundles also will have a covering. Okay, that is called as perimysium. Perimysium. And the muscle fibers also will have a covering. Okay, this muscle fibers also will have a covering. This is called as endomysium. Okay, endomysium. Got it, all of you? Yes or no? Okay, endomysium. So this is how your muscles are going to be. Okay, now students, if I remove an entire muscle fiber, okay, let's say I remove the entire muscle fiber. Okay, it won't be this thick also. This entire muscle fiber. From that, two people are going to be coming outside. Okay, two people are going to be coming outside, and one is going to be called the actin, and another one is going to be called the myosin. Okay, I'm just drawing it like this randomly, but you know, structures are not exactly like we draw sometimes. This is going to be called the actin, which is the thin filament, and this is called the myosin, which is the thick filament. So this, inside the muscle fiber, there are going to be myofilaments, and inside the myofilaments there are going to be two proteins called as actin and myosin. Okay, two proteins called as actin and myosin. Now we will go back and read NCERT, so that time NCERT will be thoroughly clear for you. Okay, thoroughly clear for you. Okay, so look here. Let us examine a skeletal muscle in detail to understand the structure and mechanism of contraction. Each organized skeletal muscle in our body is made up of number of muscle bundles or fascicles. Okay, fascicles. I think fascicles, fasciculi, you can tell. Okay, they are held together by a common collagenous connective tissue called as the fascia. Yes, clear? Each muscle bundle contains muscle fibers. That's what I told you. Now, each muscle fibers is lined by a plasma membrane called as the sarcolemma. So the plasma membrane of the muscle is going to be sarcolemma, enclosing the sarcoplasm. Okay, enclosing the sarcoplasm. That is the inside layer. Muscle fibers is a syncytial as sarcoplasm containing many nuclei. Okay, they are multinucleated. Okay, multinucleated with more cytoplasm. Now, the endoplasmic reticulum, that is the sarcoplasmic reticulum of the muscle fiber, is a storehouse of calcium ions. Students, this you have to remember and keep, because in future we are going to talk about this. A characteristic feature of muscle fiber is the presence of a large number of parallelly arranged filaments in a sarcoplasm called as myofilaments or myofibrils. What is it called? Myofilaments or myofibrils. So in the presence of a large number, in a muscle fiber there's a presence of large numbers of your parallelly arranged filaments in the sarcoplasm called as myofilaments, okay, or myofibrils. Each myofibril has an alternate dark or light bands on it. A detailed study of myofibrils has established that the striated appearance is due to the distribution pattern of actin and myosin. The light band called is because of the actin, because it is the thin filament. Okay, this is the thin filament. Now rest I'm not going to read. I'll teach you, then I'll read. Okay, this you will not understand. Now 1 second, students. Let me just show you the diagram. Okay, this is the diagram. Look at the diagram. This is your entire muscle. Okay, inside this, what you're seeing, this is your muscle bundles. Okay, muscle bundles, and muscle bundles are called your fascicles. Okay, fascicles. See here, you can see muscle bundles called fascicles. To keep them between, okay, between them you have a layer which you can call a connective called as fascia. A connective called as fascia, which is a collagenous connective tissue which is going to keep all the fascicles together. Now, students, if you see one muscle, if it is removed, okay, these are the muscle fibers, okay, these circles are the muscle fibers. So if I remove that, you're going to see many myofilaments, okay, you're going to see many myofilaments, and inside the myofilaments you will be able to see the sarcolemma, that is going to be the outside plasma membrane, sarcolemma and sarcoplasm, okay. So the plasma membrane is going to be called lemma, okay, sarcolemma, and inside layer, okay, with the cytoplasm is going to be plasma, sarcoplasma. Endoplasmic reticulum is going to be sarcoplasmic reticulum. Okay, all these terms we are going to write. Okay, now students, you can see blood capillaries are surrounding it. Obviously, everywhere muscles, blood capillaries have to surround. Okay, now before I make you understand that, let me write here. Okay, 1 second, let's drink some water. Okay, so students, here some things that you have to remember. Okay, now remember the plasma membrane of the muscle is called as sarcolemma. Okay, so students, plasma membrane of the muscle is called as sarcolemma, and the endoplasmic reticulum is called as sarcoplasmic reticulum. Okay, then the plasma, okay, the plasma membrane is done, the inside, okay, the cytoplasm of the muscle will be called as the sarcoplasm, okay, sarcoplasm, and mitochondria also is there, sarcosome, that you don't have to study. I think that's all. Okay, sarcolemma, yeah. Okay, now students, some more things that you have to know, that okay, this sarcoplasmic reticulum is a storehouse, okay, storehouse of calcium ions. Now this is going to be an important topic when you are going to learn how the muscles are going to contract, so please keep in mind. Okay, please keep in mind. Now the next thing that you have to know is, they told that, so how is the flow of this, ma'am? So muscle, okay, muscle to muscle bundle, okay, and this muscle bundle, I'll write the name, okay, muscle bundle. Inside the muscle bundle there is muscle fibers. This muscle fibers are made up of many myofilaments or myofibrils, okay, myofibrils or myofilament, okay. And this myofilaments are made up of two things called as actin and myosin, okay, actin and myosin. Clear? Yes. Now each names we will write. Okay, now students, this muscle, okay, this muscle bundles are called as fascicles, okay, fascicles, and to keep these fascicles together we have fascia. Okay, muscle fibers can also be called as muscle cells, can also be called as muscle cells. And trust me, such kind of flowchart you will not find anywhere. So make a note of it and don't be like, oh, this and why to write all of this. All this I'm writing and helping you out. Okay, muscle cells. It's neat only, no? Okay, muscle cells. Now this myofibrils or myofilaments have the thin filament. Okay, thin filament and the thick filament. Now students, thin filament, thick filament means what? One fellow is thin, okay, thin one line is there, and one fellow is thick, okay, thick filament. So one is thick filament, thin filament. These two are going to contribute in making the light and the dark bands, okay. That is why on the muscles, let's say if I'm drawing the muscle here, okay, if I'm drawing the muscle this way, okay, like this, and here you go. Muscle, you will notice that there are some dark bands. Okay, I'm drawing first the dark bands. Okay, there are some dark bands and there are some light bands. Okay, some light bands. This dark bands is constituted because of the myosin and the light bands are constituted because of the actin. Now we have to learn how does a muscle contract. Okay, how does a muscle contract? For that you have to learn a diagram, okay, which I'm going to draw. I think we can do it after a break. Okay, we can do it after a break. So I'll resume back after a break, students. I will teach you this because this is a very important concept. Okay, after that we have very small portion to understand. Okay, before that let's do one thing. Before we go for a break, I'll teach you a little about each of the proteins. Okay, then I will come to the structure. Okay, structure proteins are here. I hope I left enough space. Okay, yes. Okay, chalo, I will first teach you this one and then I will come back here. Okay, so students, when a muscle is contracting, okay, you should remember that there are some things inside the muscle. Okay, there are some things inside the muscle. So we can call the contracting, okay, the contracting unit, okay, contracting unit of muscle as sarcomere. Sarcomere. This is going to be the contracting portion. Now let's say I'm going to draw some diagrams here. Let's indicate one color. Let's take some bright colors only. So this is going to be your actin. Oh, drawing a straight line is going to be a task. Okay, let's say here is going to be your actin. This is also going to be your actin. Okay, this here also there is going to be actin and this is also going to be your actin. Okay, what I've drawn in yellow is going to be actin, so I'm writing it here, actin. Listen for 5 minutes and let me tell you, you will be mind blown that it is so easy or what. Okay, trust me, it is going to be very, very easy. Now students, I'm going to draw myosin. Because myosin is a thick filament, I am basically drawing a thick filament. Okay, thick filament. Let me just okay, I'm going to draw a thick filament. Yes. Okay, let's make it a little more thick. Full thick. Okay, this is your myosin. Okay, this is your myosin. Now myosin. Now to keep actin in place, okay, to keep actin, you have to wait until I finish drawing, okay, otherwise it's going to be little task. Otherwise you'll be like, draw it faster. I cannot draw it faster, students. I have to make you understand also. To keep actin in place, okay, to keep actin in place, there is a line which is called as the Z line. What line? Z line. Okay, it's a zigzag line. Okay, so Z line is there to keep actin in place. So this Z line, I'm going to be labeling it here also, Z line. To keep, obviously for actin, if line is there, then myosin also will get angry. So myosin also we have to give a line, yes or no. So to keep myosin in line, there is a line called as the M line. Okay, what line? M line. Myosin M, you think like that. Okay, M, so M line. is there to keep myosin in place. Now, when a muscle, see, when you think of contracting a muscle, you will never remember that this much is happening. But understand, students, whenever you are thinking of contracting your muscle, your Z line, these two Z lines, they are going to move forward. Okay, they're going to move forward like spring and all, no, they're going to push forward. So that is when your muscles are going to contract. Now, before that, there is some, I would say, like there are some bands that you have to study. Okay, bands that you have to study, and I'll teach you those bands. Okay, 1 second. I had drawn a diagram for all of you, but I think we lost it. Did we lose it? I think we did not lose it. Okay, no problem. Okay, now students, look here. This portion where only actin is present. Okay, this portion where only actin is present is called the I band. What is it called? I band. I stands for isotropic band. Iso means what? Same, right? So only actin is present, so it will be called as isotropic band. How many isotropic bands are there? 2 isotropic bands are there. You can see here. Okay, 2 I bands. I band, I band. Now here in between where actin and myosin, both of them are there, this portion, okay, this portion is called as the A band. What band? A band. Okay, A stands for anisotropic band. Anisotropic band. This is called as anisotropic band because obviously for the fact that anisotropic means not same, two different materials are there. So myosin also is there, actin also is there. Clear? Now, there are more zones to this, okay? Where only myosin is present, okay? Where only myosin is present, this portion is going to be called the H zone. It's going to be called what zone? H zone. H zone is also called as Henson zone, where only myosin is going to be present. Okay, this is the bands you have to know. Remember, myosin is going to be present. Then students, here you can see myosin is there, actin is there. It's somewhat like this. This portion is going to be called as the overlapping zone. Okay, what is this portion going to be called? Okay, this portion is going to be called, actually this portion, okay, but it's difficult to label here, so I'm just going to label here O zone. Okay, O zone or the overlapping band. O band or the overlapping band. Overlapping zone, band, anything you can tell, where both actin and myosin is there. Now these are the different types of zones. Now when a muscle contraction happens, what is going to happen? The Z line is going to move forward. Okay, when the Z line is going to move forward, there are some people who are going to reduce, increase, all of that is going to happen, which I'll teach you very soon. Okay, which I'll teach you very soon. Okay, now we'll take a break and we will come back and I will teach you, we'll continue from that thing. Okay, yes, so here Hello students, hope all of you got a good amount of break. So let's continue from where we stopped. Yes. Okay. So we are going to continue. So see, this is what we learned last. Okay, I'm just going to erase this. Chalo. Let's go here. See, this is what we learned last. So students, when a muscle is contracting, it is not like a simple process. Okay, there are some zones, there are some bands which are going to help. So the contracting unit is called a sarcomere. Now ma'am, from where to where is the sarcomere, you can ask me. From one Z line to another Z line constitutes a sarcomere. Understood? From one Z line to another Z line constitute a sarcomere. Okay. Now students, see, as I told you, when the contraction is going to happen, these people are going to come closer. Who? Z line is going to come closer. Now that you have understood the zones and the bands, let's go back to NCERT and let's read. Then you will get more idea about it. Okay. See, look here. Actin and myosin. The light band contains actin and it is called the I band or the isotropic band. Why is it called the isotropic band? Because it's a single, only same, okay, same or single, you can use the word, because only actin is present. Whereas the dark band called the A band or the anisotropic band contains, okay, contains myosin, actin as well as myosin. Both the proteins are arranged as a rod-like structure parallel to each other and also to the longitudinal axis of the myofibrils. Actin filaments are thinner. Okay, actin filaments are thinner compared to the myosin filament, hence are called as thin and thick filaments respectively. In the center of each I band is an elastic fiber called the Z line that will bisect it. Okay, a Z line that bisects it. The thin filament are firmly attached to the Z line. So students, actin is going to be caught by the Z line. So you keep that image in your head and then you try to learn this. Otherwise, it's going to be too difficult to understand. Okay. The thick filament in the A band are also held in the middle of the band by a thin fibrous membrane called the M line. Okay. So myosin is held by actin is held by Z line. Okay, myosin is held by M line. So far this is clear. Then A band is where both actin and myosin are there. Okay, where AM is there. Okay, and I band is where only A is there. Okay, only A is present. Got it? Now let's go back to the paragraph. From here, listen carefully. The A and the I bands are arranged alternatively throughout the length of the myofibril. The portion of the myofibril between two successive Z lines is considered as the functional unit of contraction and it is called the sarcomere. That's why I told you we can also call it as a contracting unit. Okay, it's a functional unit of contraction, like how your nephron is the functional unit of kidney. That means he is the one who is going to do the urine formation, the glomerular filtration, tubular secretion, tubular reabsorption. All this is going to happen because of the help of nephron. Same way here also, the functional unit of contraction is going to be sarcomere. In a resting state, the edges of thin filaments. So in the resting states, the edges of thin filaments on neither side of the thick filaments partially overlap the free ends of the thick filament, leaving the central part of the thick filament. That central part where only the myosin is present, that is called as the head zone or the Hensen zone. Okay, Hensen's zone. Got it? Now, look here students, this is the diagram. See, this is the same diagram I drew. This is the Z line. These are the actins. So obviously I'm not going to draw actin like your infinity. Okay, so instead I've drawn straight line and this is your myosin. So when the contraction is going to happen, this Z line is going to move forward. So remember one thing, that when contraction happens, okay, Okay, so when contraction happens, remember few things are going to happen. Okay, what are the few things? That is going to be okay. Hmm. When contraction happens, then Z line will move forward, or Z line moves inwards. Okay, moves inwards. Got it? Z line moves inwards. Second thing that happens is A band remains constant. A band is like, you do whatever you want to do, I am not going to move an inch. A band is going to be constant. Clear? A band is going to be constant. Okay. Chalo. Okay, A band is going to be constant. I band is going to get reduced. Okay. I band will get reduced. O zone, overlapping zone is going to be O zone, or O band is going to increase. Why so? Because when this fellow is coming together, more overlapping zones will be there. Okay, more overlapping zones will be there. Okay, head zone also will increase because actin is going to slide over myosin, and only myosin area also is going to reduce. M line is going to be constant. Okay, M line is going to be constant. These are the things that you have to remember, that when contraction happens, these are the bands and the zones that is going to decrease. So when contraction is going to happen, your Z line is going to move inwards, your A band is going to be constant, and your I band is going to decrease, and your O zone is also going to decrease. Clear to all of you? Awesome. Okay. Let's continue. And the functional unit is going to be called the sarcomere. Okay, functional unit is going to be called the sarcomere. Now, we are going to learn about the actin and myosin. Okay, structure of contractile protein. So these two proteins are the reason why contraction is going to happen. Okay, that is how our muscles are going to contract. Okay, contraction is going to happen because of these two proteins. So each actin filament is made up of two filamentous actin helically wound to each other. So F filament. So students, first remember, first actin was G. Okay, globular. It was like this. Okay, so it was called as G actin or globular actin. See here, you can see the globular actins. Then polymerization happened. Okay, polymerization happened. Later polymerization happens because of your magnesium ions, but that is not given, so you don't take tension. Because of which two filamentous structures have been formed. Okay, two filamentous F structures, F actins were formed, and on that your globular protein arranged themselves, and this is how the structure of actin is right now. Okay. Now the structure is important for you to know because there are some villains who are coming to the diagram. Okay, there are some villains coming to the diagram. So students, G actin, okay, that was called as globular actin. So each F actin is a monomer of monomeric G actins. Two filaments of another protein called tropomyosin also runs close to the F actin throughout its length. So they are telling that between these people there is one person who is going to run across the length, and that is none other than your tropomyosin. Tropomyosin. Got it all of you? Yes or no? Tropomyosin. That is the person who is going to run. Okay, now tropomyosin also runs, understood, tropomyosin runs a complex protein called troponin. Here, three proteins together, okay, three units together forms the troponin. Okay, so troponin is contributed at regular intervals, okay, regular intervals of the tropomyosin. In resting state, a subunit of troponin masks the active binding site of myosin. So students, this is a very sad story. We are going to talk about a love story which we have to make it happen. So it is very simple. Myosin, okay, has actin binding site. Myosin has actin binding site. Okay, let's say actin is the heroine. Okay, so myosin also likes actin also likes myosin, but there is a problem. Okay, now on actin there is myosin binding site. Okay, myosin binding site. Now there are villains. Okay, there are villains to the story. Okay, these are the villains to the story. First villain of which is going to be on myosin is going to be called as ATPase. Okay, ATPase. This is the villain who is going to cover actin binding site, telling that you cannot fall in love with actin. I am not going to allow that, so I am going to cover the actin binding site. Then the villain here, okay, the villain here in your actin is going to be your troponin plus tropomyosin. These two people are going to cover the myosin binding site. Is it clear? Now see, now we have to learn about the, so before I go into that, let me teach you first your actin. Okay, let me teach you actin. Okay, ad is playing, continuous ads. Yes, clear? Okay, now we are going to learn about actin. Actin. So students, this is how actin is going to look like. You are seeing a rope like structure, no, that is tropomyosin. So students, actin started forming from globular actin. Okay, globular actin, which is called as G actin. Globular actin. Globular actin. Clear? This globular actin got a little filamentous. Okay, this globular actin got filamentous and form the F actin. Okay, in that F actin, students, this troponin is going to run and tropomyosin is going to run at the entire length, and then you have troponin. Now when I talk about the troponin and tropomyosin, they are actually regulatory proteins. Students, when actin and myosin is going to come together, your contraction is going to happen. So we do not want our muscles or our muscles to contract all the time. So to regulate them, we have two people, that is called as tropomyosin and troponin. These people are going to control the contraction or regulate the contraction, because if they do not move, the myosin binding site on the actin will not be visible, and then the myosin will not come and bind. Okay, so how do this troponin work, students? Troponin structure has 3 units. Okay, troponin structure has 3 units. Let's say this is 1, okay, this is 2, and this is 3. These are the 3 units. So one is called your TP. So we can I think in your syllabus it is not there. Okay, but usually when we teach, we always teach this way. TPC. Okay, this is the one who is going to bind with the calcium. Okay, this is going to be your TPI, which is going to have inhibitory functions. Inhibitory in function, actin inhibitory, and this is going to be your troponin. So TPT. Okay, TPT. Now students, remember this. Wait 1 second. Now, this calcium is this TPC is very, very important. I'll tell you why. Now, actin diagram is clear. No, these are your troponins. Okay, these are your troponins. 3 units you can see. Each unit has one name, and as I told you, okay, one thing also is there. Now, listen to me carefully. Okay, now I'll just teach you myosin as well, and then I'll come back to this. Each myosin filament is also polymerized protein. Many monomeric proteins called meromyosin constitute one thick filament. Okay, meromyosin constitute one thick filament. Each meromyosin has 2 important parts: a globular head with a short arm and a tail. The former region is called the heavy meromyosin, and the latter is called the light meromyosin. The HHM, that is the heavy meromyosin compound, is of the head and the short arms outwards at the regular intervals, angles at each other. There is known as cross arms. The globular head is an active ATPase, okay, enzyme, and has a binding site for ATP and active site for active. So the villain on meromyosin or on your myosin is going to be the ATPase binding site. Now students, look here, this is the structure of it. Now what did they say? They told that your myosin, okay, your thick protein, is made up of it's a monomer of meromyosin. Now they told few other things. They told that this meromyosin is made up of a long tail, okay, a head region, again, okay, they wound around, and then one more head region. This is how your myosin is going to look. Now they have told some important points, like your tail region, okay, is going to be LMM. LMM means light meromyosin. Okay, light meromyosin. Light meromyosin is going to be the tail region. Then this portion is going to be the short arm. Okay, short arm or your cross arm. Okay, cross arm or cross arm. Then students, here this is going to be the head region. I think I should have written it in different colors. Too confusing in the base. One second, I'll just draw this a little above. Okay. This is one, and this is another one. Okay, now these are your actin binding sites. Okay, it's on the top only, but it is hid. Okay, it is going to be hid by your ATP binding site. This is your ATP binding site. Only if this ATP binding site will move, then only we can see the actin binding site. Okay, now let's go one by one. This is going to be your tail region. Okay, tail region, and since the tail is not a heavy region, we will call it LMM, light meromyosin chain. This portion is going to be called the cross arm. Okay, cross arm. And students, this portion, the head region, this portion is called the head region, and this whole, okay, this whole head plus cross arm is going to be called your HMM. Okay, heavy meromyosin chain. Okay, what is it called? Heavy meromyosin. Meromyosin. Okay, heavy meromyosin, light meromyosin. Now, what is exactly going to happen? See students, here also there is an actin binding site. Okay, both the persons have a binding site. That means these people will bind when contraction has to happen. But this fellow is an ATP binding site, and this fellow is a villain. Okay, this fellow is separating the hero and heroine. Okay, same way there, troponin and tropomyosin is going to separate the hero and heroine. Now, if you understood this much, remember that this meromyosin is made up of 6 polypeptide chain. Is made up of 6 polypeptide chain. 6 polypeptide chain means 4 long chains. I mean, sorry. Okay, we have to write 2 short long chains and 4 short chains. Okay, actually students, short chain, when we draw, we just draw it like this. Okay, these are the short chains. These are the 2 long chains. 2 long chains and 4 short chains. Now this is your structure of meromyosin, actin and myosin. Now the next thing is, how are these people going to meet? How are we going to move the problematic people from their head? So if I want to make sure that actin is going to visibly show the myosin binding site, then I have to remove who from it? I have to remove troponin and tropomyosin, right? They both are the villains. Okay, fathers and brothers are there. If I don't distract the fathers and brother, or if I don't tell them to move that side, I will not be able to make actin show the myosin binding site. So the myosin will understand that I have to come and bind. So for that, student, there is something called as your neuromuscular junctions. Neuromuscular junctions. Now what is neuromuscular junctions? Very simple. Very, very simple. This is your muscle. I'm drawing everything in teaching, so it will be easy to understand as well. Okay, this is your muscle. And this is your neurons. Okay, this is your neurons. Okay, this is the axon. So this portion is called as a neuromuscular junction. That means where the neurons will come and meet the muscles. Okay, neuromuscular junction. Also we can call it as your sarcotubular systems. Now listen to me carefully. This system is a little it's very easy to understand, but you have to listen carefully. So students, in the neuromuscular junction, the muscles are going to come in contact with the neurons, and they are going to give the signal telling that, okay, you have to contract your muscles. Yes or no? So if contraction has to happen, your myosin and actin has to bind with each other, come together with each other, then only your contraction is going to happen. Sarcomere contraction is going to happen. So what I've drawn here is a T tubule. What is it? A T tubule. Okay. Now when a nerve impulse is being carried, okay, let's say this is a nerve impulse, and let's say this is happening inside the muscles. This might look like a chicken leg, but it's not. Okay, chicken feet. So this is your second, yeah. Okay, this is going to be an axon ending. Now it sends a signal. Because of the signal, what is going to happen, students? Sodium influx will happen. All this you people know from neuron chapter. Sodium influx will happen, and that sodium influx will be completely toward near this T tubules. Okay, near this T tubules. Sodium influx will happen. Sodium influx will happen because of your creation of an action potential. Okay, the first step was action potential. As soon as the action potential happened, okay, the sodium influx started happening. Now inside this, okay, what happened? I think I got stuck for some time. Fine, right? Okay. OBS got just problematic. Okay. So students, here what is going to happen? Because these people are having influx of calcium, okay, lot of influx of sodium. This calcium ions from the endoplasmic reticulum, okay, ER, or your sarcoplasmic reticulum, we can say, okay, sarcoplasmic reticulum is going to release, okay, it is going to get released. All this calcium is going to get released outside. So very simple thing, action potential happened. Because of the action potential, okay, because of the action potential, second step was influx of sodium, okay, influx of sodium ion. Third step was, because of the influx of sodium ion, sarcoplasmic reticulum okay, released calcium ions. Okay, now the next catchy part. So this calcium ions, you tell me, where is it going to go? It is going to go to your actin. Okay, why ma'am? To the actin. Because don't you remember that in actin there is troponin where we have a TPC binding site, yes or no? So here we have three people, which is your troponin. In that troponin, student, there is a TPC binding site, that is a calcium binding site. So this calcium will come and bind with this TPC site. Okay, with this binding, the myosin and the tropomyosin and the troponin is going to get deviated. Okay, so there is going to be a deviation. Okay, deviating troponin and tropomyosin. Understood? So basically we caused a distraction. Now your mom is coming to hit you. What do? Immediately? You run that side, you run this side, or you go and make some noise so that your mom's concentration to hit you will reduce. So same way here also, because of the release of calcium, the troponin TPC site will bind with the calcium, and that calcium will cause a deviation in the troponin and tropomyosin, and automatically, students, the myosin binding site will be visible. Myosin binding site will be visible. Yes or no? Clear, all of you? Is it clear? Fine. So your myosin binding site will be visible. Myosin binding site will be visible. Now one job is done. Okay, we distracted one person, and because of it, one person is visible, myosin binding site. Now our next job is going to be to make sure that on the myosin head, the ATP binding site also moves. Then both of them are going to meet. Because students, very simple, on the actin, okay, on the actin, there is myosin binding site, MBS. If this is not visible, okay, if this is not visible, then the myosin cannot come and bind. Same way, on the myosin, there is actin binding site, ABS. If ABS is not visible, actin also cannot come and bind. So how does their combination happens? All of you mark your attendance quickly, click on the like button. Okay. Now students, what is going to happen? Let's say this is your myosin. Okay. This is your myosin. This myosin has to come and bind on the actin. Basically, actin is going to slide over the myosin. Now you're going to study sliding filament theory. Now listen here, this is where you are going to learn. Now students, one thing I missed to tell here is, when an action potential is created, acetylcholine, okay, a neurotransmitter called ACH, which you have studied in your neurons, okay, ACH will be released. This ACH will be collected by the receptors on the neuromuscular junction. Okay, that will lead to an influx of sodium into the tubules. Okay, sodium into the tubules. Got it? ACH will be released. That ACH will be collected by the receptors. Okay, hi Chintu. Will be collected by the receptors on the sarcoplasm, neuromuscular junction, and these receptors are going to collect it, and because of which influx of sodium is going to happen. Okay, so with action potential, you please remember that ACH will be released, because it's a neurotransmitter. ACH will be released and it will be taken by the receptors. Okay. That is the reason for the influx of sodium in the T tubules. T tubules. This you shouldn't forget, T tubules. Now we will go back and read NCERT. See, muscle contraction is initiated. Listen carefully. Whatever I taught you, keep it in mind. Is initiated by a signal sent by the central nervous system. Okay, via a motor neuron. A motor neuron, along with the muscle fibers, connected to its constitute. Okay, connected to a constituent motor unit. Okay, So motor neuron plus your muscles are going to form a motor unit. The junction between the motor neuron and the sarcolemma of the muscle fiber is called a neuromuscular junction. Understood? Okay, or a motor end plate. A neural signal reaching this junction releases a neurotransmitter called acetylcholine, which generates an action potential in the sarcolemma. This spreads through the muscle fibers, that is to the three tubules, and causes the release of calcium ions into the sarcoplasm. Increase in calcium levels leads to the binding of calcium with the subunit of troponin on the actin filament, and thereby removing the masking of the active sites of myosin. Is this clear? Okay, very easy, very easy. That what just happened, that action potential, so the signal came with the help of your motor neuron, okay, from the central nervous system, and then from the motor neuron it went into the junction. In the junction the neurotransmitter got released. Okay, neurotransmitter got released. That neurotransmitter got accepted by the receptor, and then there was an influx of sodium ions in the T tubules. From there, okay, because of that there was a release of calcium ions from the sarcoplasmic reticulum, and that led to the binding of calcium plus TPC in the troponin. This led to the distraction, and because of which myosin binding site is visible now. Now our next job is to make sure that we are making the active binding site also visible. Okay, active binding site also visible. Now let's see that. See student, that is called a sliding filament theory. Now listen carefully. This is a very simple example. There is ATPase binding site. They have already told ATP binding site is there. Now this is ATP. Okay, one ATP. Now that ATP, students, will get hydrolyzed. So the first ever step is going to be hydrolyzing ATP. Hydrolysis of ATP. Hydrolysis of ATP. That ATP will get broken down. So ATP will get broken down to what? Ma'am, we all know it is ADP plus PI. This ADP plus PI will be on the head of the myosin. Okay, head of the myosin. Then students, because the ATP went, now the myosin binding site and active binding site is visible, and they will form a cross bridge. Okay, cross bridge. So cross bridges form when a myosin and an actin bind. So here is their marriage. No, actin plus myosin marriage will happen here. Actin plus myosin marriage is going to happen here. Now students, since in the head of the myosin ADP and PI was there, it is too heavy, so this has to release. So because of that, the myosin will move its head, causing a power stroke. Okay, causing a power stroke. This power stroke is happening because ADP plus PI is rushing out of the myosin head. Okay, out of myosin head. Because of the power stroke, like this, a power stroke is happening. What is going to happen? Imagine something is on my head. Okay, something is on my head. I moved. Obviously, it's going to fall down. No, same thing is going to happen, students. Actin was there on the myosin's head. Myosin's head moved. So automatically, actin got slided. Actin moved. Immediately in that gap, another ATP will come and attach. Okay, in this gap, another ATP will come and attach. This is going to break the cross bridge. So breaking of the cross bridge. Now how the question is going to come? They are going to give numbers and they are going to ask you to label, or they are going to ask you the answer of the order. Okay, order. So students, what just happened? What happened? So very simple, sliding filament theory. Okay, what is it called? Sliding filament theory. Okay, sliding filament theory. Now let's see what is going to happen in sliding filament theory. First thing is going to be ATP will come and bind on the myosin head. So ATP plus myosin. Okay, next step is going to be hydrolyzing. Okay, hydrolysis of ATP. Hydrolysis of ATP means ATP will get broken down to ADP plus PI. Okay, now that ATP ADP plus PI will be like, since ATP went, now the actin binding site, okay, actin binding site is visible. Okay, actin binding site is visible. That actin binding site is visible, so that will lead to what? That will lead to myosin plus actin binding leading to cross bridge formation. Okay, cross bridge formation. Yes, clear? One second. Okay, done. Okay, cross bridge formation happened. Now this cross bridge will be planning to continue, but that is when your ADP and PI, okay, ADP plus PI will rush outside the myosin head, okay, outside the myosin head, and that is called the power stroke. Okay, that is called the power stroke. So students, here you should remember that, let's say this is your actin, okay, this is your actin, okay, it will bind with myosin, like this binding will happen, okay, like this binding is going to happen. So this is going to cause the cross bridge. Okay, here what is going to happen, your myosin, okay, actually I should draw it ulta, okay, this way. Okay, like this binding will happen, this is going to be your myosin, actin myosin would have bound, this will lead to power stroke. Here what is going to happen, your myosin will move the head, okay, myosin will move the head, and because of that, because that's going to be power stroke, okay, ADP and PI will go, ADP plus PI will go out, this will cause the actin, okay, this will cause the actin to have a space, okay, can you see there's a space. Now this space, who is going to take use of your ATP? ATP is fully in love, no, so ATP will run and come, ATP will run and come, and what is going to happen, students? Here again ATP will bind to myosin head. By then your action potential also would have come down, okay, so automatically what is going to happen, students? Automatically your actin binding site, and in the sense myosin binding site on the actin also will not be visible. So this is your beautiful sliding filament theory. Okay, this is your sliding filament theory. Got it or not? Okay, easy BC to us. So first step is going to be hydrolyzing of the ATP, second step is going to be formation of cross bridge, third step is going to be power stroke, where the ADP and PI will leave. That will lead to this step, students, breaking of your cross bridge. Okay, breaking of cross bridge, breaking of cross bridge. Clear? Beautiful sliding filament theory is ready here. Okay, so this is how, okay, this is how contraction is going to happen. Okay, contraction of muscles. Now let's come back here. Okay, I hope this is all clear. Okay, this is all clear. So see, these are the structures. So on the actin there is myosin binding site, which is hid by the troponin and tropomyosin. Okay, tropomyosin and troponin is going to hide the myosin binding site. Okay, it is going to mask, not unmask, mask. It is going to mask myosin binding site. Okay, myosin binding site. Same way here also, students. Okay, this fellow will have ATP. This is going to mask actin binding site. Okay, actin binding site. Done. Then when you go down, how do they unmask it? So here what is going to happen, students, when an action potential is generated, that means I am sending a signal to my brain telling that I want to contract my muscles. So immediately what will happen, the neuromuscular junction, that is the gap junction between the muscles and the motor neuron, that junction will understand this. Okay, this fellow wants to, or this girl wants to contract a muscle, and they are going to release, or they're going to send the signals, and that signal will have acetylcholine in it. That acetylcholine will be accepted by the receptors which is present on the neuromuscular junction. That receptors will create an influx of sodium. Okay, action potential means automatically influx of sodium will happen. That influx of sodium will go through the T tubules, and that will let the sarcoplasmic reticulum release the calcium. Since inside this the calcium levels are going to keep increasing, automatically this calcium is going to bind with the TPC site. Okay, TPC site on the troponin, and that will allow the troponin and tropomyosin to deviate, causing the unmasking of myosin binding site. Now, to make the actin binding site visible, what we have ATP, who is the villain sitting on the myosin head. So ATP hydrolysis we will do, that will lead to the formation of ADP plus PI, and this ADP plus PI will allow actin to bind. Okay, so actin binding site will be visible, forming cross bridge formation. Now cross bridge formation will happen, children. That means myosin and actin's marriage will happen. Now what will happen, this ADP and PI will be like, I don't want to sit in myosin's head, I want to go out. So they will move forcefully, and that will cause a power stroke. Because the power stroke happens, what will happen, the myosin head will move. When the myosin head moves, the actin which is attached to it will get separated. In that gap, ATP will come and again bind with the myosin head. This will lead to breaking of the cross bridge. Okay, breaking of the cross bridge. Hi Tanuja. Clear to all of you? Yes or no? So, no issue, no issue. Okay, yes. This is what has happened so far. Okay, so far we have understood this much. Okay, clear? Chalo. Now we will go further. This is your sliding filament theory. Okay, now here they have written a little about it. See, pulls the attached actin towards the center. The Z line attached also pulls. So here, students, they're talking about the shortening. As I told you, how the muscles are going to get shortened and who are the people who are going to reduce. Okay, so as I told you, the first process of shortening of muscle will be Z line moving inwards. Okay, Z line moving inwards. First ever step, if a muscle has to contract. Okay, oh please take care, dear. Okay, Z line will move inwards. Inwards. Okay, you have to recover before the Navratri series, so take enough rest. Yeah, okay, take enough rest. By the way, let me check how much attendance is happening on our Navratri series. Okay. Okay, that I was just checking that. Okay. 1 second, students. Okay, I was just checking how many attendance happened. Okay, how many monkeys marked attendance. Okay, yes, ready? Chalo. So here, first thing, Z line is going to move inwards. Second thing that is going to happen, students, the I zone, sorry, I band. Okay, I band is going to decrease. Okay, next is going to be I band will decrease. Okay, then A band is going to be constant. Okay, A band is going to be constant. It's like I don't care what is going to happen, I am not going to move. Same way, one more stubborn person is going to be M line. Even M line is going to be constant, okay, or unchanged. Then students, O zone or your O band is the only fellow who is going to increase. Okay, who is going to increase. Why? Because O zone is an overlapping zone. So when something is coming forward, let's say I don't have 3 hands here, I wanted 3 hands to show this, but yeah, this is your myosin, this is your actin. Now Z line is pushing actin. Okay, Z line is pushing actin, so more overlapping zones are going to come, yes or no? So O band is going to increase. Okay, O band is going to increase. H zone, that is your Hensen zone, where only myosin is present, is going to decrease. So exactly this is what happens when contraction is going to happen. See, you can read it here as well. It is clear. See, Z line attached to these actins are also pulled inwards. See, I told you Z line will be the first one who is going to pull inwards, thereby causing shortening of the sarcomere. That is, contraction is going to happen. It is clear from the above steps that during shortening of the muscle, contraction of I band is going to happen. Okay, gets reduced, whereas A band will retain the length. The myosin that is releasing the ADP plus PI goes to its relaxed state, a new ATP will bind and the cross bridge is broken. So it is going to happen, everything is going to happen simultaneously. That is, cross bridge formation will happen. Cross bridge formation means what? Myosin actin binding will happen. Immediately action potential will be enough. I don't know that I want to contract my muscle. Now I'm like, don't need, you just relax, yaar. Okay, so that time what will happen? Action potential will get over, calcium binding will reduce. Automatically, when calcium binding reduce, what is going to happen, students? The troponin and tropomyosin is going to go back, and no more myosin binding site will be visible. Same thing will happen here. When myosin is going to a relaxed state, ATP will come and bind back. Okay, so this is going to happen. Everything is going to happen simultaneously. Don't think that, okay, after one decade of life it is going to happen. Everything is going to happen tuck 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 tuck. Okay, everything is going to happen tuck 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 tuck. Okay, clear? Yes. Okay, tuck 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 is over. Chalo. Now we are going to understand. See, here you can see. Okay, this is how a muscle will look when it is relaxed. Okay, everything is going to be proper. Like head zone is there. This is where only myosin is going to be there. Okay, only myosin is going to be there. This is the I band where only actin is going to be there. Okay, this is the full A band. Okay, where both myosin plus actin is going to be there. Okay, AM is going to be there. This is the Z line, this fellow. See, now contraction is happening. That means myosin and actin has met each other. Okay, cross bridge formation is happening. Now the Z line will move inwards. Okay, Z line will move inwards. When the Z line is going to move inwards, students, you can see what and all is decreasing. Now students, maximally contracted, like when we're doing exercise in the gym, full contraction is happening, right? We try to push as much as possible. So same way, maximum contraction has happened. So here you can see full shortening has happened. Okay, now one more question they can ask you is, one sarcomere, okay, one sarcomere will have how many bands in one? Okay, one sarcomere. So say 1, 2, 1, say this is one sarcomere, one Z line to one Z line. What and all bands are there? 2 bands are there, A band and I band. So one full A band is going to be there, and plus half I band. Okay, half I band. Okay, plus one more half I band. Why am I telling half and half? Because of a symbol funda. The funda is nothing but, see students, Z line is there, so this is going to be the I band. Okay, so this side I cannot take. I can only take from this Z line to this Z line. So that is going to make it half. Okay, same thing goes here. I band is going to be from here till here. I cannot take the full. I have to take half. So half I band, I band. So a normal sarcomere, one sarcomere, okay, one sarcomere will have one A band, okay, plus one I band. Clear, all of you? Yes or no? One A band plus one I band. Done. Clear? Okay, done. Clear, all of you? Sure. Okay. Got it. Now we will go to next topic. Okay, let me thank you, Chintu. I think, Chintu, I have seen you on NEET English. Okay. So much work is there, students, because October 11th is very special for us. So much work is there for all of us. Okay. Chalo. Okay. Yes. Now we are going to learn some skeletal systems. Okay, skeletal systems. Ready? Okay, here we go. Skeletal systems. Okay, now look here, students. So this chapter is divided into two parts. Okay, I'll just write that here. Yes, Chintu, I know you. If you comment once also in my class, I'll never forget you. Okay, that is how I am. I always read comments and I see the names. So once at least if I've seen your name somewhere, I will usually never forget a student. Okay, because they're taking their time to comment, so I find it very precious. So yes. Okay, students, this chapter is divided into two parts. Okay, locomotion and movement. So my regular Vedantu NEET English students like Tanuja, Soni, Harshine, okay, Ratis, all of these are like my kids only. Like I can scold them like my kids because every day I'm seeing them. No, they're like my kids now. Okay, like every day I get I'm like, okay, they come to class. If they don't come to class, I'll scold them, and so they'll become like my kids. Okay, so that's the truth. Oh, that's great. So locomotion and movement is divided into two parts, students. First is going to be learning about all the different kind of locomotions. Okay, all the different kinds of locomotions. Yes. Then with locomotion you are studying about how muscle contraction happens. Okay, but muscle plus muscle contraction. Okay, muscle contraction. Students, you want any topic from the previous for me to revise? Just tell me. Chintu, Tanuja, what are you listening? Okay. Yes. So muscle contraction is there, students. Then you learn about your sarcomere, which is a functional unit of contraction, and then you will learn about actin and myosin in depth. Okay, actin and myosin. Okay. Then we learn the most important question. Always question keeps coming from sliding filament theory. Okay, sliding filament theory. Done? Okay. Then we will learn about contraction done, sliding filament theory done. Now, second part of this chapter is going to be a skeletal system. Okay. In this, you have main two parts. Okay. Axial, appendicular. Okay. And then you have disorders, and then boom, done. Okay, you learn about the joints, and then you learn about the disorder. This is how much this chapter is there. Okay, so let's see how much we can do today. Rest we will do tomorrow. Oh, tomorrow I have Navratri series plus DNA, so I'll not find time tomorrow, students. Some other, like after Saturday. Okay, Friday is very jum day for us because something big is coming. So yes. So skeletal system consists of a framework of bones and a few cartilage. This system has a significant role in movement shown by the body. Imagine chewing food without jaw bones and walking around without limb bones. My God, scary, right? Bone and cartilage are specialized connective tissue. This we have learned where in structural organization. The former has a very hard matrix due to the calcium salts in it, and the latter has slightly pliable matrix due to the contractin salt. So students, here what are they talking about? They're talking about two people. That is going to be your bone. Okay, in your bone we know that we have osteocytes. Okay, osteocytes, and they are filled with calcium. Okay, calcium. Second, they are talking about cartilage. And we have learned in structural organization that they are made up of chondrocytes. Yes or no? Chondrocytes present in a lacunae. Okay. Now, in human beings, this system is made up of 206. Okay, 206 bones and a few cartilage. It is grouped into two principles. Okay, Tanuja, are you going to tell Soni and Ratis and Harshain and all of them that Gopika ma'am called us her children? Tanuja? They'll come tomorrow and they'll be like, ma'am, seriously, ma'am. And then the other ones will be jealous. Okay, other ones will start fighting. Telling that, ma'am, even I attend regularly. You never told my name. Okay. So they will start full doom jam. They'll fight. Okay, Rani will fight. Then who else is there? Roshni will fight. Okay, some of the names that I can remember. Chintu is already here. She's not started fighting. So yeah, Tanuja, are you going to do that? Okay, so now students, so our entire skeleton system is going to be 206 bones. Okay, out of this your axial skeleton is going to be 80 bones. Okay, distributed among the main axis, that is the skull, vertebral column, sternum, ribs, joints. The skull, okay, that we are going to come to. Okay, now 1 second. Okay, before I go into this, so students, here I'm going to write few things. Yes. So your entire skeletal system, okay, skeletal system has how many bones? 206 bones. 206 bones. In this 206 bones, we can divide them into two categories. Okay. One is going to be called your axial skeleton. Okay, axial skeleton. One is going to be called your appendicular skeleton. Appendicular skeleton. Got it? Now, axial skeleton, how many bones are they telling, students? Axial skeleton, they are telling 80 bones. Okay, and appendicular skeleton, how many bones will be there? Appendicular skeleton, all of you calculate and tell me. 126 bones. Okay, 126 bones. Now first I will teach you what are present in these, Okay, very good, Chintu. What are present in each of them, I'll teach you. See students, this one starts with your cranium. So axial skeleton means this part of your body. Okay, appendicular means this part of your body, your hands and your legs. Okay, now axial skeleton consists of your cranium. Okay, cranium, that is your skull, your facial bones. Okay, facial bones. ear ossicles, hyoid. I'm coming from top to down. Sternum, ribs, and vertebral column. Yes or no? These are the parts of your axial skeleton. Now each of them, how many bones are there? Cranium has 8 bones. Okay, facial bones are 14. Ear ossicles are 6. Okay, 3 each. Hyoid is 1. Sternum is 1. Ribs are how many, students? All of you tell me in the chats how many ribs are there? 12, 13, 14. Okay, how many ever you want you can tell. Okay, how many ever you want you can tell. Tell me how many? 7 true ribs are there, 8, 9, 10 is false ribs, 2 floating ribs, 12 are there. Yes or no? Shall I write? Okay, 12 ribs are there. Okay, vertebral column, students, we have 7 cervix, okay, 12 thorax, 5 lumbar, and 1 tailbone. So it is going to make of around 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25. Yes. Okay. So 24. So 12 plus 12. Okay, here you write 12 plus 12. That is going to be 24. Okay, or 12 pairs you can write. Okay, 12 pairs you can write. This is going to be 26 bones. Okay, 26 bones. Done. All of you. Chalo. Now this is the calculation. Now calculate and tell me if 80 is there. Okay, let me also take my phone and calculate. I always calculate. Okay, whenever my students are doing, when I calculate. Because sometimes when we write hurry bari, sometimes we will miss out on something. 14, okay, plus 6, plus 1, plus 1, okay, 30, plus 24, okay, plus 26. Perfect. We have 80 bones exactly in your axial skeleton. Okay, exactly 80 bones in your axial skeleton. Now we can learn about axial skeleton. Okay, now that you know what is your axial skeleton. So students, if I have to draw a skeleton, so this is the cranium. Okay, this is going to be the most scariest skeleton you have ever seen. Okay, and let's say here you come, that is going to be your hyoid bone, and then when you come down, you will see your sternum. Okay, Tanuja, I asked you something, Tanuja. I think you weren't here that time. Okay, and then this is going to be your ribs. Okay, ribs are going to be there. This is going to be the funniest skeleton actually, and then your vertebral column will be there in the back. Okay, so this makes your axial skeleton. Okay, but appendicular means what, students? Hands, okay, legs, okay, pectoral girdle, pelvic girdle. Okay, this makes your system. So got it? I'm going to erase this because it might haunt some student who is going to see this notes. So we'll erase it. Okay, chalo. Now that you have understood, we will go one by one. The skull is composed of 2 sets of bones. Okay, one is going to be cranium bones. Okay, bones of your skull. Bones of your skull or the brain box, and second is going to be bones of your facial bones. Okay, facial bones. Okay, now skull, cranium. So cranium, students, to remember, there is a trick. Okay, cranium bones, there is a trick to remember. What is the trick? Step off. Okay, step off. S stands for sphenoid. T stands for temporal. E stands for ethmoid. Guys, if you want my full lectures every day for every chapter, you can follow me on Vedantu NEET English. Also join my Telegram. Telegram name is Biology by Gopika. Okay, you will get all my class links on this Telegram, and also you will get my personalized notes links where I write short notes, where I write, you know, GM Express, that is Go Become Am Express, and all my GMAs are in this Telegram channel. Okay, so this is my Telegram ID. You can join this Telegram ID and you can watch my lectures on Vedantu NEET English Vedantu NEET English channel. Okay, Vedantu NEET English. Now this is going to be the channel. Now E stands for ethmoid, P stands for parietal. You will get all the tricks, you will get all the easy ways to learn it, so definitely follow. Okay, O stands for occipital. Okay, and F stands for frontal. Students, in this, who is paired? Who is paired? Tell me. Tanuja madam is so busy. From that time I'm asking that I asked you a question, but she's not. I think she's going, and I think network issues there. Okay. Okay, step off. Tell me who is paired. Out of the students, out of the 8 bones, okay, cranium has 8 bones. In that, 2 are paired. 2 are paired. Okay. 4 are, the rest are unpaired. Okay, rest are unpaired. So in this, PT is paired. Okay, who are paired? P and T. PT teachers are always 2 in number. Yes or no? You have always noticed one boy will be there, one girl will be there, or 2. No, it's usually a lady teacher and a gents teacher. Okay, PT teachers or any training for that, PT teachers will always be in a pair. So that's how you remember. Now students, how do you know the cranium bones? This is your frontal bone where you always get headache, no, like that. So this is your frontal. Okay, this ear portion is your temporal. This is your parietal. Okay, backside is your occipital. Sphenoid and ethmoid you cannot see. If I do a cross, you will see this is your sphenoid. Okay, ethmoid is going to be somewhere in the below your orbitals of your eyes. Okay, so here you can see how the bones are arranged. See, look here, frontal. Okay, frontal, sphenoid. You can see sphenoid here. Okay, in the sphenoid, student, there is a small depression called sella turcica. Okay, sella turcica. This sella turcica will have your pituitary gland. Okay, pituitary gland. Students, I'm so sorry. Can I just get my charger and come? Okay, my phone is running out of charge and I will not be able to see your chats. Just give me 1 minute. I'll be back in 2 minutes. Just revise the names. I'm back. Just give me 1 minute, students. Yes. Done. Okay, did you all learn it? So who is going to be the ones who are going to be paired? It is none other than your temporal and parietal. Okay, temporal and parietal is going to be the two people who are going to be paired. Temporal and parietal. Okay, parietal is this, temporal is this. Okay, temporal is this. So these two are going to be paired. Now students, when we talk about the facial bones, okay, you have to talk about the facial bones. Okay, yes, about the facial bones. See, so here you come. Okay, now cranium and facial bones, that totals of 22 bones. How are they 22 bones? 14 plus 8 bones. Okay, now cranium bones are 8 in number. They form a hard protective covering called cranium for the brain. The facial region is made up of 14 skeletal elements which form the front part of the skull. The single U-shaped hyoid bone is called the hyoid bone, is present at the base of the buccal cavity. Okay, this is not needed. It is also included in the skull, is not needed. Okay, clear all of you? Yes or no? Okay, that okay. Each middle ear. Now students, slowly, so we have come, now we have to learn the facial bones. Okay, we are going to go back to that slide and learn the facial bones. See, facial bones are around 14 of them. So what are the facial bones here? See, this is going to be parietal, okay, temporal, occipital, occipital condyle will come. Now I am going to mark everything in red. Those are your cranium bones. Okay, those are your cranium bones. So you understood, no? This sphenoid bone has a depression called as sella turcica. In that, pituitary gland is going to be there. Now this is that. Now you see which are the bones of your facial bones. Okay, so you can start with your nasal. Okay, nasal bone. So this is going to be your nasal bone. Okay, lacrimal bone, which is near your eyes. Then zygomatic bone, students, which is your cheekbone. When you blush and smile, no, you smile and you'll put your makeup. This is your zygomatic bone. Okay, then you have maxilla, mandible. Okay, maxilla and mandible is the hyoid. You can see it's at the base of the cavity. It's not actually a part of the skull. Okay, so now here some of the bones are actually missing. There is palatine, vomer and all. So I'll only make you write it. Okay, I'll only make you write it. Ready? You'll write with me. I have to see the chats here, okay, because charging is happening. So that's why I'm looking this side. Don't be like, why is this ma'am looking that side? 1 minute, 1 minute. Okay, okay. Okay, clear. Oh, neck tissue is there. Okay, 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 okay. Fine, fine, fine. No problem. Now, students, let's write here. Okay, let's write the bone. So, first one is going to be your nasal bone. Nasal bone. Two of them. Okay, nasal bone. Yes. Then your infranasal bone. Okay, infranasal bone. So that is also inside the nose itself, infranasal bone. So where I'm keeping the specs, no, there you have your nasal bones. Okay, where I'm keeping the specs. Then you have infranasal bone. Then you have your cheekbone. Okay, cheekbone, which is called the zygomatic bone. Zygomatic bone. Okay, two of them. Two of the bones. Then students, there is some more bones which are not mentioned. Lacrimal bone is mentioned for you. Okay, lacrimal bone, two of them. Then palatine. Okay, palatine and vomer. This is two. This is one. Okay, then you have maxilla, and we have maxilla is 2. Then we have mandible. Okay, mandible, which is 1. Got it? Now count and see 14 are there. All of you count and see 14 are there. 2, 4, 6, 8, 10, 11, 12, 13, 14. Very nice. 14 facial bones are there. Okay, 14 facial bones are there. Is this clear? So nasal bone, infranasal bone, cheekbone, zygomatic bone, okay, lacrimal bone, then palatine and vomer is present, students. One vomer is going to be here, okay, palatine is inside. Now palatine will be near the eyes. Then you have maxilla and mandible. Okay, in this you have to see which are paired and which are single. 2 people are going to be single, that is going to be vomer plus mandible. Okay, vomer and mandible is going to be the singles of the group, others are all double. Okay, others are all double. Got it? Done. Okay, facial bones done. Now when we come further, okay, here I think here only have to write. Okay, when we come further, students, you have to remember that there is hyoid bone. So write it in a different color. Hyoid bone, which is going to be present at the base of the buccal cavity. Okay, only one fellow U-shaped bone this is. We also call it tongue bone because of the shape. Okay, tongue bone, U-shaped tongue bone, base of the buccal cavity. Okay, then we have ear ossicles. What do we have? Ear ossicles. Ear ossicles, 3 of them are there, students. Stapes, incus, okay, and malleus. Stapes, incus, and malleus. Now if you want to remember their names, you have to remember Miss Has. Miss Has you have to remember. So M stands for malleus, this is hammer shaped. Incus, anvil. Stapes, stirrup. This has become ulta. Okay, ulta I wrote, otherwise it will be Miss Has. Okay, Miss Has. Very good, Chintu. You know all the tricks and all. Very nice. U-shaped tongue bone. It is present in the base. So now, students, this is 1. Okay, this will be 3 each. Okay, 2 years you have no, so 3 each is going to be there. Now, when we come further to the vertebral column, before going to vertebral column, I'll teach you the sternum. Okay, I'm only teaching you. I'm not reading this. Okay, sternum. Now sternum, students, is a dagger shape. Okay, but for me it looks like a tie. Tie is there, no? We used to tie to school, office people tie, no? This tie, it looks like a tie for me, like this, like this. Okay, now doesn't it look like a tie? So this portion, okay, this portion, see now usually sternum 3 portions, it's not mentioned in your NCERT, but when I'm teaching I usually teach. Okay, this is called as your prosternum. Okay, prosternum. This is going to be your mesosternum. Okay, mesosternum, and this is going to be your sphenoid process. Okay, this is going to sternum. This sternum is present ventrally. Okay, okay, fine. This is present ventrally, students. To this only your ribs are going to attach. Okay, like this your ribs are going to attach. To the ventral side, it is going to attach to the sternum. I have taught you in breathing, no? And to the what is that, dorsal side is going to attach to the thoracic vertebrae. Okay, thoracic vertebrae. Now this is clear, no? Now this is your sternum. This is going to be one bone. Okay, one whole bone divided into 3 parts, but it's okay if you do not remember prosternum, mesosternum, and sphenoid process also, it's okay, but just remember sternum is there. Okay, next is going to be your vertebral column. So vertebral column, students, first portion is going to be called your cervical vertebrae. Cervical vertebrae will have C7. Okay, 7 bones will be there. C1, C2, C3. In that first 2 bones you have to remember. Okay, first 2 bones you have to remember. The first bone, okay, first bone is going to be called the atlas. Atlas. Okay, second bone is going to be called the axis. Atlas and axis. Okay. After that, you can just label it like this: C3, C4 up to C7. Next portion is going to be a thoracic vertebrae. This is going to be the largest, T12. This is going to start from T1 up to T12. Okay. Lumbar is going to be L5. Okay. Lumbar is going to be L5. Then sacrum and coaxial, student, they are fusion. So sacrum will constitute around 5 bones which are fused, and coaxial will have 4 bones which are fused. Okay. Together they'll form the tailbone. Together they'll form the tailbone. Clear? Yes or no? So in kids 33 bones will be there, in adults 26 bones will be there. Okay. All of you tell me if it's clear or not. Okay. Now this is your vertebral column. Okay, vertebral column. Next is going to be your ribs. Now students, in ribs, very simple thing is going to happen. What is going to happen? No, so I'll show you the image, then I'll show you the. So look at this image. You will see that 1 to 7. Okay, how is a true rib true rib? Wow, what is this? How can I call a rib true rib? Okay, how can I call a student true? Okay. Thank you so much, Chintu. Your telling itself is a very sweet thing. Okay, thank you so much. What's your actual name? Chintu looks like one this name, cartoon name. Okay, so what's your actual name? Okay, students, look here. If I want to call something true, that means they should be true, they should be loyal. Now, if Chintu is my true student, Chintu forever is going to respect me as a teacher, and Chintu will always make sure that she gives me or he gives me the respect that is required for me, right? Same thing will happen if it is going to be a rib. Now, if I want to call a rib, okay, if I want to call a rib, true rib, I will have some criteria. First criteria, okay. Okay. Now true ribs, very fancy name. I will call a true rib if it is going to ventrally attach to sternum, and dorsally attached to the thoracic vertebrae. This is when I will call a rib true. Okay, true rib. Yes? He, ma'am. Okay. So now listen carefully. If I want to call something true, it has to ventrally attach to my sternum and dorsally attach to my thoracic vertebrae. Okay. But if it does not do this, I'm going to call it the false rib. So out of the 12 ribs, students, okay, out of the 12 ribs, 1 to 7, okay, 1 to 7 ribs are called as the true ribs. 1 to 7 are called true ribs. Why? Because they are attaching dorsally and ventrally correctly. Now 8th, 9th and 10th are called false ribs. Why? Because simple factor that ventrally they're not attaching to the sternum. Okay, ventrally they are attached to the hyaline cartilage, of the 7th rib. Okay, they are attaching to the hyaline cartilage of the 7th rib. Hyaline cartilage of the 7th rib. Instead of attaching directly to the sternum, they are actually attaching to the 7th rib. Okay, so this is why I call it false rib. Then the last set is going to be the 11th and the 12th rib, which I'm going to call as the floating ribs. Why am I calling the floating rib? Because they are not at all attached to anything ventrally. Okay, ventrally, no attachments. Okay, so basically they are floating. Now we will go back to the diagram. You will understand thoroughly. Look here, students. You can see sternum, I mean the ribs are attaching ventrally, dorsally also. Okay, here also you can see up to 7th one. Okay, up to 7th one it is attaching properly. 8th one, can you see how it is attaching? It is attaching to the hyaline cartilage. See, it is attaching to the 7th rib. Okay, 9th one also is doing the same thing. Okay, 10th one also is doing the same thing. Okay, here you can see all these people. Okay, all these people. So they are attaching to the 7th. Here you can see the connection got over here. Now you see 11th and 12th, they are floating. Okay, they are floating. They are not at all attached ventrally. Okay, ventrally not at all attached. So we will call it the false ribs and floating ribs. Clear? Clear all of you? Any doubts so far? Now, students, since these ribs, okay, since these ribs are attached ventrally and dorsally, they will have two condyles. Okay, to attachment, two condyles will be there. So we can call them bicephalic. Bicephalic. Clear? Bicephalic. So let's go back to NCERT little. Okay, now look here. Students, one more thing I want to tell you is about your vertebral column. About your cranium. Okay, about your cranium, students. So in the cranium there is occipital. No, occipital is the last one here. That fellow is going to have dicondyle. He is going to have two articulating substances. Okay, we call that articulating substances as condyle. This condyle substance is going to articulate with the vertebral column. Okay, it is going to articulate, so it will be called as dicondyle. So look here, the skull region articulates with the superior region. Okay, superior region of the vertebral column with the help of two occipital condyles. So we call it a dicondylic skull. Understood? Two condyles are there. Now our vertebral column is formed by 26 serially arranged units called the vertebrae, and it is dorsally placed. It extends from the base of the skull and constitutes the main framework of the trunk. Each vertebrae has a central hollow portion called the neural canal through which the spinal cord passes. First vertebrae is called atlas and it articulates with the occipital condyle. So you can remember that if the condyle has two, okay, occipital condyle has two of them, who is going to condyle with them? Atlas. Okay, who is going to articulate? Atlas is going to articulate. Okay. Atlas, it articulates with the occipital condyles. The vertebral column is differentiated into cervical, 7 of them, thorax 12, lumbar 5, sacrum 1, coccygeal 1. So we consider them 1-1 fused. Okay, 1-1 fused. The number of cervical vertebrae are 7 in almost all mammals, including humans. The vertebral column protects the spinal cord, supports the head, and serves as a point of attachment for ribs and musculature of the back. Sternum is flat bone on the ventral midline of the thorax. Okay, ventral midline of the thorax. Here, students, ribs also same thing is written. See, there are 12 pairs of ribs. Each rib is a thin flat bone connected dorsally to the vertebral column, ventrally to the sternum. It has two articulation surface, so dorsal end, and hence it is called as bicephalic. Okay, on its dorsal end, and hence it is called bicephalic. Now, first 7 pairs dorsally they are attached to the thoracic vertebrae, and ventrally they are connected to the sternum with the help of a hyaline cartilage. The 8th, 9th and the 10th pair do not articulate directly with the sternum but joins to the 7th rib with the help of hyaline cartilage. Okay, with the help of hyaline cartilage. Now these are called vertebro-condyle, that is the false ribs are called vertebro-condyle. The true ribs are called vertebro-sternal. Okay, because they are connecting to the sternum. No, vertebro-sternal. This is called as vertebro-condyle because they are connecting to the cartilage. Okay. Last two are called as floating ribs because they are not at all connected. They are called as floating ribs. Thoracic vertebrae, ribs and sternum together form the rib cage. Form the rib cage. So who and all? Vertebrae. Okay, vertebrae plus ribs plus sternum together is called the rib cage. Then 3 of them together is called the rib cage. And this is how the rib cage, ribs plus rib cage is going to look like. I hope I haven't missed anything. Okay, I hope I haven't missed anything. I'm just going to check my notes as well, students. Where vertebrosternal, you don't forget. Okay. There's a foramen magnum and aperture in the spinal cord, which is going to help for the articulation and passing of that. Okay, I think everything is done. Okay, everything is done. So that's it for today, students. That's all for today's class. So we have done up to, I'll just show you the notes recap. Okay. Okay, so we started with the introduction. Okay, with the introduction, and we understood about different kinds of movements done by different types of cells in our body. Okay, amoeboid and ciliary. Okay, here I've written a complete overview, so you will always get an overview of my class. So here you can see muscular, skeletal, neural system. All these three have to work for your movements to happen. Muscular movements are going to be of your limbs, jaws and tongues, and amoeboid movement is between your WBC and macrophages, where diapedesis will happen. Cytoskeleton also movement will happen, that is of your microfilament. Ciliary movement is happening because of the lining of the trachea and bronchi, lining of the fallopian tube, and it helps in the movement of the ova. Flagellary movement is happening because of the help of sperm. Then students, based on the location, we have 3 types of muscles. That is going to be a skeletal muscle, cardiac muscle and smooth muscle. So skeletal muscle characteristics is here, smooth muscle characteristic is here, and cardiac muscle characteristic is here. Okay. Then here I have written the 4 properties of muscle, and based on location how they are categorized. Okay, excitability, elasticity, extensibility and contractibility. I think students will think that this is some other point by itself if I change the color. So I'm writing it here, excitability. Okay, excitability. Thank you so much. Yes. So students, excitability, elasticity, extensibility and contractibility. So based on the location, you have 3 types of muscles: smooth muscle, skeletal muscle and cardiac muscle. Okay, and here I have drawn the muscle. Okay, one second. Yeah, here I've drawn the muscle to make you understand about each of the part. So we have fascicles. I always write this spelling wrong. Okay, fascicles. Okay, fascicle, they are the muscle bundles, muscle fibers, then the covering, endomysium, perimysium and epimysium, then your myofilaments, how the actin and myosin is coming out. Then here students, I have written about the different names of each of them: plasma membrane, endoplasmic reticulum, cytoplasm, muscle, muscle bundle, muscle fiber, myofibrils, actin and myosin. Then I've written about the contracting unit, that is called your sarcomere. From one Z line to another Z line, it's called a sarcomere. I've written the different labelings here as well. You have understood the labelings, I hope. Then I have told you about each of the actin and myosin. Okay, here you have myosin. Here you have actin. Here you have how the troponin is going to get deviated, leading to the myosin binding site to be visible. Here you have about the sliding filament theory, which is one of the most important concepts. And here I've taught you about the neuromuscular junction, again sliding filament theory. I've taught you about what happens when contraction happens. Okay, and here the same thing, locomotion. I've told you about the chapter. Then I've taught you about axial skeleton. And here you go. Okay, the cranium I have given. Okay, facial bones. I'll just write it here. The students will not understand. Facial bones, all 14 of them I've written. Then I've written about all the sternum, okay, cervical, yes, and ribs. Okay, so everything is done and dusted, students. Thank you so much for joining. See you all in next class. Okay, I hope all of you enjoyed. Okay, all of you enjoyed. Yeah. Thank you so much. Thank you so much. Okay, so thank you. See you all in next class. Okay, guys, don't forget to join October 11th. Okay, October 11th, I will see you guys on Okay, I will see you somewhere soon, that is on Vedantu. Those students who are preparing for boards, please do join this channel called Vedantu Boards and KCET channel. Okay, do not forget to join. See you all on October 11th. Thank you so much for joining. See you all in next class. Bye.